Hello everyone, and welcome to this audio podcast today. This audio podcast is part of the cytometry cyclone. This audio podcast is part of the Rakas and Bombard episode, and this audio podcast is part of a Storm Robotics production. Continuing on then, and looking at the importance of IL-2, interleukin-2, IL-2, interleukin-7, IL-7, and interleukin-15, IL-15, and co-stimulation in human kidney transplantation or renal transplantation. A series of biological a series of biological agents that inhibit or block the action of inflammatory molecules, particularly tuber necrosis factor alpha or TNF alpha, have been utilized for pharmacotherapeutics of autoimmune conditions, and through another immunological approach. Most of the sort of historical therapeutics or agents directed at cytokine receptors had targeted the interleukin-2 receptor alpha chain. This approach is based on investigations that interleukin receptor alpha is not conveyed by normal quiescent cells or quiescent cells, but it is articulated by abnormal T cells that are implicated in AAR or acute allograft rejection incidents. Targeted interleukin 15 or IL-15 seems to be important amongst the literature. Targeting interleukin 15 or IL-15 seems to be important because it is primarily involved in the inflammatory stimulus for tuber necrosis factor alpha synthesis, and is evident and evidence indicates and highlights that it is implemented across autoimmune disorders and conditions. The autoimmune maladies that are an initial focus for interleukin 15 directed pharmacotherapy include those where there is confirmed elevated serum levels of interleukin 15 or IL-15. Major effects have also demonstrated the evaluation of cytokine IL-15 as pharmacotherapeutic agent in recipients with renal cell or kidney cell tumors and metastatic or malignant melanoma, as well as an adjuvant component of cancer vaccinations. In one investigation, cardiac allografts have been observed under anti-CD25 treatment or pharmacotherapy, and the investigators had identified, for example, that with pharmacotherapeutics, around 29% of the cardiac allograft recipients had rejected the allograft in use of anti-CD25 regimen. One laboratory investigation had established that macrophages secreting T cell growth factor, interleukin 15, IL-15 are abundantly involved in rejecting cardiac allografts, thereby suggesting that interleukin 15 or IL-15 could be substitute for IL-2, interleukin 2 in anti-CD25, a monoclonal antibody in anti-CD25 monoclonal antibody blocked rejection. The investigation commenced. The investigation commenced to assess the viability of interleukin 2 and interleukin 15 to induce T cell proliferation of anti-CD25 block T cells. The investigation had involved sort of measuring and identifying the frequency of interleukin 2 and interleukin 15, IL-2 and IL-15 in peripheral blood and anti-CD25 treated recipients during cardiac allograft rejection and from healthy control of volunteers, and both the absence and presence of mice or murine anti-CD25 monoclonal antibody BT563. The investigators had informed that interleukin 15 or IL-15 may be a decent substitute for an interleukin 2 or IL-2 in rejection process amongst the rejection process in recipients with anti-CD25 cardiac allograft with anti-CD25 cardiac allografts, and in some similar works, investigators, at least one team, one team retrospectively, had also hypothesized that differential expression of interleukin 2 and interleukin 15 receptor subunits on cycling T cells in vivo may direct activated T cells to respond on interleukin 2 and interleukin 15, thereby regulating the homeostasis and expression of interleukin 2 and interleukin 15 receptor subunits. The team had investigated whether T cells express the interleukin 15 IL-15 receptor alpha chain, which may render cells responsive to interleukin 15 or IL-15 during initial cell divisions. The alpha chain for interleukin 7 IL-7 receptor was not detected on dividing T cells in vivo. Laboratory data had suggested that blocking interleukin 2, IL-2, or IL-15 signaling may differentially affect T cell expansion in vivo. The group, the investigators had used a CFSE cell trace labeled lymphocytes from interleukin 2 deficient mice laboratory model as part of laboratory models, and the investigators had informed and demonstrated that the gamma chain down regulation required a T cell activation and a cell cycle progression, as well as interleukin 2 signaling. In the absence of interleukin gamma chain, where it is expressed at high levels, and where blockade the gamma chain is expressed at high levels, and where blockade of interleukin 2 or IL-2 receptor and gamma chain downregulation is precluded or inhibited, again in vivo on cycling T cells. That draws this audio podcast to a close. This audio podcast is as part of the cytometry cyclone. This audio podcast is part of the Rakas and Bombard episode, and this audio podcast is part of a Storm Robotics production.